Okay, next up we have John Lagergren from NC State University, and he'll be talking to us about biologically informed neural networks, guide mechanistic modeling from sparse experimental data. So go ahead, John.
Thanks. Yeah, so similar to John Nardini, this talk is also slightly different than what's on the SciM website. There's still a lot of overlap. You guys already saw from the previous talk about using neural networks for the automatic differentiation from noisy data. And this builds on that by incorporating some domain expertise, in particular biological information. So let's get started. Let me try to, okay, there we go. I'm going to start with the application. So our application is studying collective migration, and that simply refers to the coordinated movement of individuals or populations. And this field of study has lots of applications in like cancer progression, animal development, pedestrian dynamics, so on. And in the field of cell biology, a really popular experimental protocol used to study collective migration is a scratch assay. So a scratch assay involves first growing a cell monolayer up to some desired initial cell density, and monolayer just means that the cells are side by side and not on top of each other. And once they've grown to some initial cell density that you want, create a scratch down the center of the monolayer. And then over the next 48 hours or so, you can take imaging data as the cells start to converge back into this empty region. And using either manual cell counts or a computer vision algorithm like the tracer that Erica was talking about earlier, you can aggregate individual cell counts in these vertical columns shown on the left and get 1D spatiotemporal data across your different time measurements or images that you've taken in time to create these data surfaces. And so scratch assays, they're popular for a reason. They're highly applicable in a variety of contexts, including cancer modeling, wound healing, tissue engineering, regenerative medicine, and so on. And so a key challenge in this field is to, for both mathematicians and scientists alike, is to pair mathematical models with the spatiotemporal data in order to discover the underlying mechanisms that describe the temporal evolution of these systems. And this is done generally using reaction-diffusion models. So in this example, for a cell density U, which is a function of x and t, its time, for reaction-diffusion models describe its time evolution in two parts. There's a diffusion term and a reaction term. So in the diffusion term, that's the first one, dux, quantity x. D here is a diffusivity function, and then the reaction term describes proliferation or cell growth with growth function G. However, throughout the literature, there's a lot of different opinions on how to pick the diffusivity and growth functions mechanistically. So starting with diffusivity functions, for example, you can have the diffusivity function be simply constant, or you can have it be increasing with cell density U. It can be linear if the exponent m is set to 1, or nonlinear for other values of m, and k here is the carrying capacity. And they can get more complex than that. So diffusivities 3 and 4 describe different behaviors as the cell population increases in cell density. So the first case describes where cells become more densely packed. This encourages diffusion so that they can move away from each other. Where the fourth one predicts the opposite, where due to cell adhesion, the higher the density, the less diffusion there is. So there's a lot of opinions floating around on what diffusivity functions to use. Similarly for growth functions, most of the time it'll be the logistic population growth model with intrinsic growth rate R and carrying capacity K. But the disagreement in which mechanistic functions to use here for D and G, that motivates our use of equation learning methods, specifically to uncover what the appropriate diffusivity and growth functions might be. So equation learning, John already talked about this. So basically the first big assumption is that the measured data arise from some dynamical system. And in this case, we assume that the dynamical system takes the form of a PDE, this unknown function f, And that describes the time derivative of your quantity of interest, in our case, cell density U. And of course, we assume appropriate boundary and initial conditions. But more than that, there's assumptions on the observation process, in which when you make an observation, in this case, U hat sub ij, that just means that the cell density at spatial point xi and time point tj. So that's a function of the solution of your P to E, Uij, but it's also obscured by this error process. And we like the generalized least squares statistical error function, especially in biology, because this can describe proportional error very, very well, as you saw in John's talk. And it's made up of a few components here. So there's the epsilon term, which we assume is just IID, standard normal distribution. But then that's scaled by some noise level sigma, and then it's proportional with the population values themselves, this U sub ij. And gamma here, we're treating as a hyperparameter, and that refers to the proportionality constant. And you can note here, if gamma is 0, then this is just mean squared error, or least squares. And when gamma equals 1, that describes a fully proportional error model, and so on and so forth. And the objective overall for equation learning is, given these noisy observations you had, we want to uncover or discover the functional form of F, our unknown PDE. So John just talked about sparse regression. So I'm going to focus on an alternative popular equation learning method called physics-informed neural networks. So this is work from Maziar Raisi and George Kardianakis at Brown University. And so what this method does is, say you have noisy data and you have some unknown dynamical system U. So you first use a MLP, it's just an artificial neural network, a multilayer perceptron, to approximate the solution of the PDE. And that's just done by training it to approximate the noisy data, as you saw in John's talk earlier. However, you can use automatic differentiation to construct both the left-hand and right-hand sides of some governing PDE. And then, if you jointly optimize both the parameters of your PDE on the right, so in this case, D and G, as well as the weights and biases of the neural network UMLP, you can use this joint optimization process to not only fit the noisy data, but also parameterize some pre-specified governing partial differential equation. So this is an extremely powerful idea. However, in our case, since there's a lot of discussion and debate on the function or the mechanistic forms of D and G, since these are unknown, physics-informed neural networks are limited here because they do require the pre-specification of the right-hand side of the PDE that we're interested in. So this is where our work comes in with biologically informed neural nets. So it's an extension of physics-informed neural networks. In the bottom here, this is the exact same picture from the last slide. You have your surrogate model U, and using automatic differentiation, you can piece together this partial differential equation. However, because D and G, the diffusivity and growth functions, are unknown, we replace them with neural networks. And in this case, we're just calling them DMLP and GMLP. And both of these are functions of the cell density U, which is also being approximated by the surrogate model UMLP. And using the same joint optimization procedure, you can optimize both the neural network for U against the noisy data measurements that you have, as well as learning what D and G should be by satisfying the right-hand side, the left-hand, right-hand sides of the PDE. And additionally, there's an additional loss term here in the middle, L constraint, which I'll talk about, but this accounts for any domain expertise or physical or biological knowledge that a modeler can include in this optimization process. And finally, the last step of this, because what we're presenting here is an equation discovery method, is once D and G have been trained, they can be visualized to guide the selection of a new mechanistic model, and we'll talk about that. So biologically informed neural networks, or BINs, they're trained using this multi-objective loss function. So L total is the total loss, and it's made up of 3 components. So first is the generalized least squares error component of the loss function. And as you saw in John's talk here as well, this is just least squares in the numerator. And then it's to account for the statistical error model, we also divide by the cell densities U raised to the gamma. And then added to that is the PDE error. So in this case, these are automatic differentiations of the neural network outputs and D and G. And you can subtract the right-hand side from the left-hand side, square all of those terms, and by driving this entire loss term to 0, the right-hand side learns to match the left-hand side. And in that way, you're discovering the nonlinear forms of D and G. And finally, we know that diffusivity and growth have to fall into some biologically meaningful ranges. So for example, diffusivity, if that lies in between some D min and D max, any values of D outside of those values, you can penalize that with this soft constraint here, D squared. Similarly for derivatives, say you assume that diffusion is an increasing function. Well, any partial derivatives of U with respect to its input, the cell density, any partial derivatives of D with respect to U, the cell density, that are negative, you can also penalize those by the square and add them to the loss function. And the same thing for the growth function G. So ultimately, we want D and G to learn realistic dynamics. So there are 2 key steps here to make that happen. First, just from standard protocol and training neural networks, we want to prevent overfitting. So first step is to, when you optimize the parameters of these networks, use mini-batch optimization with the gradient-based atom optimizer. We also partition the noisy data into training and validation sets, in which the training data is used to update the parameters, and then your generalization error is monitored using the validation set. And finally, once the validation error stops improving for some number of iterations during training, we use early stopping to prevent overfitting to the training set. So that prevents overfitting of the neural networks, but that's not enough. We want to ensure that D and G also generalize well to the data. So we resort to a more traditional method and freeze the weights. So we freeze the parameters of D and G, and we replace the diffusivity and growth functions of the governing PDE with these multilayer perceptron models, and then numerically solve the resulting reaction-diffusion equation using the method of lines approach that John was also discussing. So let's talk about the data real quick. This is real-world data from a real-world experiment of scratch assays. So there are 6 in total. Each data set has a varying initial cell density starting at 10,000 cells per well up to 20,000 in increments of 2,000 cells per well. And these data are sparse. So like John mentioned with his few time points example, in this case, we have 5 time points, and each time point is distributed over 37 spatial points. And an example is shown on the right for the case with 20,000 initial cells per well, where the blue curve, it represents the initial condition right after the scratch has been performed. And then as time goes in, you can see the cells start migrating into that scratch. And this is the data set that we'll be focusing on rather than all 6. I'm just going to demonstrate our method using this data set, and then we'll have some error tables at the end. So for the reaction diffusion system here, recall that we replaced the diffusivity and growth functions with MLPs, so DMLP and GMLP shown in the equation here. This is what they learned over that data set. So you can see that diffusion is largely constant across cell densities, and the growth function GMLP is approximately linear and decreasing. And when you forward solve the PDE with DMLP and GMLP substituted for the diffusivity and growth functions, this is the solution that you get. And so first thing to note is that D and G are actually interpretable. You can see that one of them is constant, one of them is linearly decreasing, and these are just approximate because multilayer perceptrons as functions, they're similar to polynomials. So we're not looking for anything exact. We want qualitative behavior here. So then these exhibit interpretable parameter dynamics. However, when you look at the solution of the PDE, you can see there's some model discrepancy, especially at early time points. So in areas of high initial cell density, high cell density and low cell density, there's persistent model discrepancy here. So taking a closer look at that, we noticed that the discrepancy is mainly with this orange curve. That's the first time point at 12 hours. And in areas of high cell density, so the box on the left, we can see that the curve is over-predicting for that time point. And similarly at low cell densities in the second box, the one on the right, the curve at the first time point, 12 hours, is also over-predicting. So these two MLPs, the D and G MLP, those are universal function approximators. They're similar to polynomials. So they are learning from the set of continuous functions of the cell density U. And this model discrepancy shows or indicates that perhaps density-dependent diffusion and growth, that assumption may be insufficient to capture the true dynamics of the system because we have this model discrepancy here. And in particular, because of this overestimation at early time points, where the later time points look pretty good, this suggests that there may be a time delay that scales the magnitudes of the diffusivity and growth specifically for early time. So to implement this idea, we're going to introduce a time delay function, also a neural network. So script T, we're going to replace that with a neural network T MLP. That's a function of time. And we're going to update the diffusivity and growth functions, where diffusivity is now assumed to be a separable function of this time delay function TMLP that multiplies DMLP. And DMLP, it's the same multilayer perceptron as the previous case. It's a function of cell density U, and similarly for the growth function. And so when you plug these into the equation, TMLP is just a function of time, so it can be pulled out of the x derivative. And so this is the resulting PDE that you get. So in the inside of the parentheses here, you have just the reaction diffusion equation with diffusivity and growth replaced by D and G MLPs. And then the whole equation is scaled by this new time delay function. And the TMLP, in this case, we chose it to have a sigmoid output activation function, and that just ensures that all of the outputs of that neural network are between 0 and 1. So it literally does just scale the diffusivity and growth as a function of time. So using our updated PDE, we train a new set of bins on the same data set, and again, decently interpretable parameter dynamics here. So diffusion is increasing, it's non-zero at 0 cell density. Similarly to the last time, GMLP is linearly decreasing, and TMLP performs exactly as we sort of predicted by being smaller for early time and then converging to unity for later time. And when you take these three neural networks and forward solve the PDE, you can see that, all right, interpretable, we just covered that, virtually all of the model discrepancy here is removed. So in particular with this first time point at 12 hours or 0.5 days, this orange curve now fits the actual dynamics very accurately, including with later time points as well. So, like I mentioned, there are six data sets in total, and we trained a bin on each one of them with this updated partial differential equation model. And so now you're looking at the DMLP, GMLP, and TMLP solutions for each data set, where the colors now correspond to the different initial cell densities used for those. So some things to note here. For the diffusion function, or for the diffusivities, we have some non-zero baseline diffusivity even when cell density is 0. And then most of them are increasing and concave up. As far as growth is concerned, these are all approximately linear and decreasing, except for the 10,000 initial cell case, which we're going to treat as an outlier. And finally, the time delays are all sigmoidal, these S curves that scale the diffusivity and growth down for early time, about during the first 24 hours or so, and then converge to 1. So using these visualizations, we want to posit a new mechanistic model, because the whole goal here is to discover a new equation. And so that's what you're looking at here. This is our posited model. So you can see that now these terms don't have the MLP in them. So this is just on the inside of the parentheses of this PDE, you have the reaction diffusion equation with diffusivity function E, growth function G. And then that whole equation is scaled by this time delay function t. So I'm going to walk you through how we generate or how we posit each one of these terms. So starting with diffusivity, you see that the non-zero diffusivities at cell densities, even zero cell densities, implies that there's some baseline diffusivity d naught. And then to account for the increasing and concave up trends, we're using the porous Fisher diffusivity function, where the diffusion coefficient d multiplies u over the carrying capacity k raised to some m parameter. And that accounts for the concave up as well as increasing trends that we see in the data. The growth functions, because they're all linearly decreasing, this strongly suggests that the logistic population growth model is correct. So we're going to go with that as well. That's the intrinsic growth rate R, also carrying capacity K. And finally, the time delay function, because it's sigmoidal, we're just going to use the logistic regression function with that adds two new parameters, beta naught and beta 1. So now that we have a posited PDE model, that's a mechanistic model, we can now do a investigation using traditional techniques, in particular inverse problems. So we can estimate the unknown parameters in order to minimize the generalized least squares error. And when you do that using a traditional parameter estimation approach, this is the solution of the mechanistic PDE that we just posited, which fits the data remarkably well, almost as well as when the diffusivity growth and TMLP or time delay functions were replaced with MLPs. So that's good news. However, this is just one data, skipping ahead, sorry. So what we want to do is compare our model to the current state of the art. So the current state of the art involves two equations. One's the classical Fisher KPP model shown here, and this uses a constant diffusivity function and logistic population growth. Then the generalized porous Fisher model, the diffusivity function here, so it also uses logistic growth, but the diffusivity function here is d times this u over k raised to the m power, also present in our posited PDE. And the bin-guided delay reaction diffusion model that we're positing, that's just the terms written explicitly into the equation. Okay, so each one of these models, we did the inverse problem, estimated the parameters, and then forward solved the final PDE to get these mean squared errors using the generalized least squares statistical error model. And unsurprisingly, the model that was predicted by the biologically informed neural networks has the smallest error. And I say unsurprising because it's a much more complex model. So with more parameters and more complexity, of course, it can fit the data better. So we need an additional metric that can balance the model complexity with the error. And so we're going to look at AIC scores, or Akaike information criterion. And in this case, lower is better. And across all 6 data sets, except for one with 14,000 initial cells per well, the model that we've posited based on what the neural networks learned has a better AIC score compared to the current state of the art. And in particular, the difference in scores and the difference in errors is greatest for large initial cell densities. So mathematical modeling is one part, but if it doesn't tell us anything about the relevant biology, it's only useful for predictions. So the interpretability part, in particular with why our model seems to perform better at large initial cell densities, there's 2 sets of biological motivations that actually support including this time delay function that we've discovered. So first, when the scratch is performed, it's unsurprising that cells are damaged along the scratch. Now, cells, they move around, they communicate with each other. So damaged or dead cells at the border can inhibit these normal cell functions and even physically block, because the cells are in a monolayer and they're just side by side. They can, the damaged cells can physically block otherwise healthy cells from migrating into the center of the scratch where there's low cell density. Another biological motivation comes with changes in density-dependent cell functions. So cells tend to differentiate when approaching carrying capacity, and differentiation means that they can lose their ability to diffuse as well as proliferate into new cells. And so high density populations may contain fewer of these mobile and proliferative cells, and so that could motivate why there's a observed time delay in the time it takes for the cells to start diffusing and proliferating into that scratch zone. So, in conclusion, the basic 3 take-home messages from this work: we've introduced a new equation learning methodology that extends physics-informed neural networks by replacing the actual terms of the unknown PDE with neural networks themselves. And this is very useful for biological applications, especially where the dynamics are highly nonlinear and you don't have a lot of data and you have a lot of noise in your data, as we showed with this real-world experiment. Talking about real-world data, most of these methods are illustrated using simulated data, and we also use simulated data just to make sure that the MLPs could learn. If we actually knew what the diffusivity and growth functions were, we ensured that they could indeed learn what those forms are. But the key part in this work is that we did use this on real-world data. And finally, using this approach, we discovered a so far unconsidered novel time delay term, and this can have important implications for wound healing and cancer modeling, where this time delay or this delayed population response isn't considered. In the future, uncertainty quantification is a very important field, and obviously we didn't have any UQ in as far as the UMLP, DMLP, GMLP, and TMLP outputs were concerned. The physics-informed neural nets were recently extended to Bayesian neural networks. And by replacing each of these 4 neural networks with their Bayesian counterparts, that could be a reasonable first step into quantifying the uncertainty in both your approximation to the solution of your PDE, that's U, as well as the parameter network outputs for D, G, and T. And so we discovered this time delay term, but there's a lot more nuanced dynamics going on. So further analysis into this particular data set or scratch assays in general. This includes using the images from the beginning of the talk to construct 2-dimensional cell density profiles rather than 1 dimension, to add a second degree of complexity into these spatial interactions that cells go through, as well as investigating the mechanisms for heterogeneous populations in which damaged but not dead cells at the borders of the scratch interact with the population. So I'd like to thank you for your time. Of course, this work wasn't done alone. Big thanks to John Nardini, Kevin, and our collaborators, Ruth Baker and Matt Simpson. This work is supported by the NSF. And if you're interested in more details, we just put our preprint online last week. So you can Google biologically informed neural networks and find a 30 or so page paper that covers all of the implementation details and more analysis. And also all of the code that's been implemented in Python using the PyTorch deep learning library. All of that's been put online for you to download and play with if you're interested.